gray with intermittent points of detachment. The black is the subarachnoid space with cerebrospinal fluid. The pink here is the pia mater. To orient this correctly, be careful. Right now it's upside down. The anterior median fissure always gives you your correct orientation. Posterior median sulcus. Anterior, lateral, posterior, white matter, white funiculus, or white column. Anterior, lateral, posterior, gray matter. Now this little tiny hole here is the central canal. One of the things we learned at the beginning of this section is that there is CSF there, and it goes down around, through here, down around the bottom of the spinal cord, and then back up around the brain. So it's circulating in this pattern. And I warned you that there was a picture in the book on this circulation. It is on the exam. You're going to have to then tell me this is a dorsal root ganglion, which is sensory or afferent. This is a motor or efferent root, and we should have integration or association within the spinal cord and within the brain. Okay? All right, I'm not going to use this model. It's good to say it's too tiny. All right, I'm going to use the brain. I don't know which one. We're all talking about it right now. We're getting ready to set up our exam. Should I decide to use the brain for the control cranial nerve, I'm going to have to block out the numbers. But I'm not going to ask you for a number. I'm going to ask for the name. Olfactory, optic, ocular, motor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, accessory, hypoglossal. I really do like this brainstem, but it's missing the olfactory. Other than that, they're all there. But on the brainstem, also, I could ask you for the midbrain, pons, and medulla. Now, in the lab, I did not give you the origin of many of the cranial nerves. You can see them on here. You can see them. In the lecture, I'm going to ask you for one of those pathways I'm telling you now. Or I'll put a pathway and I'll leave some part of it blank in the lecture. But in the lab, you just got to identify everything. All right, what kind of model is this? This is a ventricular model, and we have spaces in the brain, and they're not air-filled as our sinuses are. They're full of CSF. The little pink areas are the choroid plexus, and you have one pink area for every ventricle. Lateral 1, 2, 3, and 4. Because the special capillaries and the cells within the pink choroid plexus make the CSF, secreting it. So go back and look at your mid-sagittal view of the brain and the circulatory route of this fluid. From here, cerebral aqueduct, 4, and right here would be your central canal, this little hole right here. So essentially, our brain and spinal cord is sitting cushioned within this fluid called cerebrospinal fluid.
Are you going to use that model?
Am I what?
Are you going to use that model?
I see that I am. And I probably will even need to use one of the pictures. It will depend on what stations I end up using. Okay, for the circulatory system. Now, notice how I don't have any of those models down here. Not one of them is on the exam that I know, even though I'm still working on it this weekend. So I would really, if I were you, come over here and take a look. You may be asked to tell me spinal cord versus brain. Inner gray, outer white, and the H is spinal cord. Outer gray, inner white is the brain. Simple. No surprises here. But here is much more difficult. For the circulatory system, I'm going to ask you, identify this cell. Alejandro, if I ask you to identify the cell and you know it's a neutrophil, the next question is going to be, what percentage is it found in circulating blood? What percentage are neutrophils found in circulating blood?
67.
Very good. Use the percentages that I gave you. All right, and I'm telling you now, this is on the exam. So you might want to read. Also what's on the exam is, identify this blood vessel. So what I'm going to do is I'm going to put the pointer on the diagnostic feature of blood vessel. So, Glinder, what did I tell you was the diagnostic feature of a blood vessel? Does anybody remember?
What was the question?
Norman? What is an identifying structural feature of a blood vessel? So this?
Arteries are thicker than veins.
Yep, so I'm going to have the pointer always on. I'm going to try and put it on the tunica media, unless it's too thin. You are going to come to this microscope and we're going to say, aha, I've got 4 or 5 layers of smooth muscle. This has to be a vein. The lumen is open, there's blood cells here. But if you have 1 to 3 layers of smooth muscle, and you can see that the endothelium is starting to fold up on itself, you know you have a vein. And then I could really get that. I could put a capillary in one of these pointers. And they're going to say, oh my God, there's a space in one dot. And you're just going to say, doggone her. No, you're not, because you're here today. You're going to say, I've got a lumen, I've got one endothelia or two endothelial cells, I've got a capillary. I know exactly what this is. There will be no smooth muscle. All right. Okay. Then we went to special senses, and I am not kidding you. I have put both of these models on and this on the exam. Because although we do have a few slides, these are actually a little bit better. The slides are a little bit hard to tell unless you're a spelling. Okay? So I'm going to ask you about all 5 senses. So that means that for gustatory and olfaction, I'm going to go to either the pictures from your lab manual, okay, the pictures from your lab, or I'm going to put it in the first 2025 in the form of a verbal question. Okay? So you know to focus on these for models. Okay. The respiratory system. Yes. I'm going to have one of these larynx models on the exam, and I'm going to put the bronchial tree. Now, for both labs, I did this. Okay. Together, I already know this. You tell me, what is this? What's this?
Primary.
Okay, what's this? What's this? What's this? What? Terminal. And this one here? Respiratory? And alveolar. Somewhere in there I'm going to have a tag that identify this part of the bronchial tree. But then, I'm telling you now that you have learned so much this semester, and you know that the capillaries are lined by an endothelium. And that endothelium extends into the inner wall of the heart. Right now, Anna and Maria are looking at the heart. Simple squamous epithelium makes up the endocardium. That simple squamous epithelium makes up the alveoli. And I could put a slide here and ask you to identify this space in alveoli, identify the membrane lining this space. You could tell me it's simple squamous epithelium, or I'm going to put a point that I'm going to say, what is the function of this? You have to know this for physiology, the exchange of gases. And I'm telling you now, this is sandbox. Wait till you get to phys. And you see that you have a human chemistry course before you. Also, you have to know the position, the macroscopic position of the lungs. In the pleural cavities, in the thoracic cavity, lateral to the pericardial cavity in the heart, superior to the diaphragm. We have 2 membranes, the visceral and the parietal pleura. There should be no doubt here. Now, the lymphatic system, we have a little problem in that because of the system and the way it is, I'm going to have to probably use a diagram of the lymphatics from your lab manual, and I really particularly like the histology. You need to tell me something about the organs or the cells. So I'm going to use mostly the pictures from the lab manual. The last thing is heart. Now, I have on this lab exam, you have to know the flow. And it's really an incredible thing that we have this lab exam first, because you're going to say right atrium, tricuspid valve, right ventricle, pulmonary semilunar, pulmonary trunk, pulmonary arteries to the lungs, back at the pulmonary veins, left ventricle, bicuspid valve, left atrium. Left is the bicuspid, the ventricle, then to the aortic semilunar valve, and then you're going to tell me right brachiocephalic, left common carotid, left subclavian. Told you to learn the major branches. And then, either on one of these models, depending on how many I get for this test, I'm going to go after the right and left pulmonary arteries and their major branches. And so, there is a picture in here. I showed you the picture in lab, and I said, you need to learn this. So you're going to need to know your branches. Here it is. Thanks. Can I borrow that? Yeah. The branches, there's 2 major branches of each, pulmonary artery, pulmonary vein, because you guys all know that we don't want to die of a heart attack, and the number 1 organ that gets oxygen-laden, nutrient-laden blood is going to be the wall of the heart. And then, of course, Anna, what blood vessel recollects all of the blood from the wall of the heart and takes it back to the right atrium? What blood vessel? Ivan?
The coronary sinus.
Yeah, you guys have got to know that. There's no doubt, okay? And then, of course, the cardiovascular system I'm going to cover in detail. The heart, its circulatory routes, blood and blood vessels, their anatomy, how you identify them, and then, of course, I will put the disorders on the lecture exam. There'll be no disorders on the lab exam. That's it for all 5 weeks. Do you have any questions for me?
Are you going to test us on the ACON with her?
You know what, I'm thinking about that. Either that icon up there or a picture of this, okay? Possibly. A good question. Any other questions? All right, and I'm so sorry, you guys only have till 10 to 10 because this instructor is going to be here today with her class. So,